Discussions regarding the neuroscience of perception and the psychedelic state typically center on the 5-HT2A receptor. It is often described as the brain's master switch, acting as the primary gateway to altering consciousness. However, neurotransmitters do not operate in isolation. When the brain releases serotonin, that molecule interacts with at least 14 distinct receptor subtypes across the nervous system. Located alongside the 2A receptor in the same neural circuits is the 5-HT1A receptor. It serves as the biological counterweight to its more famous sibling. While 5-HT2A is an excitatory accelerator that increases cortical activity, 5-HT1A is an inhibitory brake. Its physiological function is to quiet neurons. This specific receptor is the target of non-sedating anti-anxiety medications and acts as the internal governor of psychedelic intensity. Mapping how the brain actively calms itself through this mechanism is essential to understanding the mechanics of mood and consciousness. To understand how the same serotonin molecule produces both excitation and profound calm, we have to look at the signaling pathways beneath the cell surface. On the left of this membrane, serotonin binds to the excitatory 5-HT2A receptor, which couples to a GQ protein. This triggers a cascade that pulls positively charged ions into the cell. This influx of positive charge raises the internal voltage, a process called depolarization, pushing the neuron towards its firing threshold. On the right side, the 5-HT1A receptor couples to a different intracellular machine, the GI protein. The GI cascade causes positive potassium ions to exit the cell. As that positive charge leaves, the internal voltage decreases. This voltage drop, or hyperpolarization, moves the cell away from its firing threshold, preventing the neuron from sending an electrical signal. The serotonin system is a balance between these competing protein signals, which determine whether a cell fires or remains quiet. Beyond the individual cell, the behavioral effect of 5-HT1A depends on its location. It is found in the cortex, the hippocampus, and the amygdala. In these areas, the receptors are postsynaptic, meaning they sit on receiving neurons and quiet them, dampening activity in the brain's fear centers. This mechanism is utilized by medications like buspirone, which ease anxiety by directly triggering these calming receptors. A different role exists down in the Raphe nuclei of the brainstem, which serves as the production site for the brain's serotonin. Here it acts as an autoreceptor, sitting directly on the neurons that produce and release serotonin. When serotonin levels rise, these autoreceptors detect the increase and hyperpolarize the parent neuron, halting further release. This allows 5-HT1A to function both as a local anxiety brake in the cortex and as a negative feedback thermostat for the entire brain. The autoreceptor mechanism explains why SSRIs block serotonin reuptake within hours, yet patients typically do not experience mood improvements for several weeks. On this graph, the first dose of an SSRI causes synaptic serotonin levels to spike. However, the presynaptic 5-HT1A autoreceptors detect this sudden increase and respond. They hyperpolarize the Raphe neurons, throttling serotonin production and largely neutralizing the drug's initial effect. Raising serotonin transmission requires weeks of continuous exposure, which eventually forces these autoreceptors to desensitize and stop resisting the drug. Only when this feedback loop is overcome does serotonin transmission finally increase enough to lift depressive symptoms. The multi-week therapeutic lag is the time required for the 5-HT1A brake to desensitize. This cellular logic also applies to the physiological response to classic psychedelics. While 5-HT2A is the catalyst for the experience, classic tryptamines also bind to the 1A brake. Stimulating the 1A receptor with buspirone before psilocybin attenuates visual hallucinations. Conversely, blocking the 1A receptor with pindolol before DMT amplifies the subjective effects. 5-MeO-DMT represents a distinct outlier, possessing a massively high affinity for this 1A brake. Because its pharmacology leans toward 5-HT1A, 5-MeO-DMT often lacks geometric visuals, producing instead a formless sense of dissolution. Consciousness is shaped by the ratio between excitation and inhibition. To understand how the mind accelerates, one must understand how it applies the brakes.